This presentation explores plant anatomy, focusing on dicot and monocot structures and secondary growth. Gain insights into plant tissues and their fascinating growth processes. Dicot stems feature endodermis, vascular bundles in a ring arrangement, medullary rays, and a parenchymatous pith with spacious intercellular spaces. Dicot vascular bundles are arranged in a ring, conjoined, open, and have indulged protoxylem, aiding efficient transport. Monocot stems have scattered, closed vascular bundles with sheaths, sclerenchymatous hypodermis, and prominent parenchymatous ground tissue. Dicot leaves display an epidermis, mesophyll, palisade and spongy layers, and vascular bundles supporting photosynthesis and transport. The leaf epidermis has a protective cuticle. Stomata are more abundant in the abaxial epidermis, enhancing gas exchange. Palisade cells are elongated and vertical. Spongy cells are loosely packed with intercellular spaces aiding photosynthesis and respiration. Veins of varying thickness form the reticulate venation. Vascular bundles are surrounded by thick bundle sheaths. Monocot leaves have stomata on both epidermal surfaces, and undifferentiated mesophyll supports photosynthesis efficiently. Bulliform cells on monocot leaves absorb water, controlling leaf culling to minimize water loss during stress. Parallel venation in monocot leaves supports similar-sized vascular bundles, facilitating efficient transport across vertical sections. Secondary growth, distinct from primary growth, increases plant girth through specialized meristems in dicot plants. Vascular cambium and cork cambium drive secondary growth, producing essential secondary vascular tissues. Vascular cambium forms xylem and phloem. Initially patchy, it develops into a complete concentric ring. Intrafascicular and interfascicular cambium combine to create a continuous cambial ring for secondary growth. The cambial ring generates secondary xylem inward and secondary phloem outward to bolster plant structure and function. Cambium produces more secondary xylem than phloem, with primary phloems crushed and primary xylems intact centrally. Secondary medullary rays, formed by the cambium, allow radial transport between xylem and phloem. This slide highlights the progressive stages of secondary growth in plant stems, enhancing plant anatomy understanding. Recap: primary structures and secondary growth lead to increased girth and structural complexity, highlighting plant growth intricacy.